Hello everyone. We are going to look at the GOLD webpage. The GOLD stands for Genomes Online Database. This is described by the paper that you can find in the discussion board, and I think it's one of the papers assigned for this week. And part of the paper kind of describes the website. So I thought it would have been easier to go through the website with a screencast. So if you go on the webpage here, genomesonline.org, and I think the link is on the paper too. You will see that, first of all, the numbers are slightly different than we would expect from reading paper. The numbers are higher, so they have a bunch of more samples of genomes that they described actually in the paper. If you look on the left side, there is the menu with a bunch of options. Some of them are pretty interesting. So if you look at the genome map, over here, we can see that all of these numbers, all of these little dots, that describe how many samples were actually obtained from that region. We can, yeah, okay, Chrome works. We can kind of travel around, look at where they are. We can actually zoom in some of them. Let's look at these in the Pacific Ocean. Of course, my mouse doesn't work now. Looks like it's, okay, my mouse is not really working. Anyway, the more you zoom in at some point, it should just simply show you narrow down the number of samples that were found, and it will tell you which sample you are actually looking at. In the meanwhile, I'm going to try to reload the page. That's a very nice feature because it allows you to see geographically where certain microbial communities are and what they are, and when they've been analyzed and so on. I'm gonna try to reopen this page. In the meanwhile, another nice function. There we go. Let's see if this works. Let's go. No, it doesn't want to know. Anyway, I'm gonna close this page up. Another nice function is the search function. Of course, we might have a clear idea in mind about what we want, and here is where we can follow that path. We can select different fields based on what we want to look for, and we also have different kind of searches. We have a normal search, advanced. We have metagenome search. Here's advanced search, metagenome, and metadata search. I'm just simply going to look for I don't know, Escherichia coli, just to look how that actually works. So if I write it, Escherichia coli, and I'm gonna submit my search. Probably misspelled it, so I'm just gonna look for coli. That's probably easier. We can see, there we go. We can see that it opens a map, and if my map works, it should show where the samples were taken. And over here we have the organism name, the domain is a bacteria, the type, so they did whole genome sequencing, and the project status. Here I have lots of them are incomplete, some of them are permanent draft, and so on. And some of them are actually completed and published. I want to look at those that are completed and published, for example. Let's say I want to look at this one. This was complete, and I'm going to click on that. It opens a new page, and it brings me to the description of the sample. I can even collapse the empty field so I don't get confused, let's say. Here is the domain, the genus, species, and so on. The specific strain I found. They say that it's also public. I want to remember that some of the data here are also private, so they're not publicly available. And the person who did it and the contact for that person, when it was done. Here there are links to the NCBI. If you look at the bottom left of the screen, when I pass over it, it opens a new page. Well, I can actually open it for you. And it will send me to the taxonomy browser and the NCBI. Then it's a sequencing status complete, and the whole size, how many chromosomes, GC content, a little bit of statistics, and you should actually, let's see if it sends to a comprehensible map. Okay, error the information. Oh yeah, so based on the block, hopefully we will be able to use some features of the website. I had some problems today too, so well, sorry about that. Anyway, It says where it was isolated from, the locations in the state of New York, Ithaca, and basically lots of other data about Escherichia coli, about this specific sample I was looking for. So this can be done for every sample we're looking at. That is very interesting. So let's go back and let's try to go back, use another kind of search. Let's use a metagenome search. Okay, here I have lots of more options, as you can see, and I can look at the study, either studies or as samples. I can get a study name based on the project ID, if I have it, and so on. I'm just going to leave it like that for now. I can look at the ecosystem. This is a logical search, if either matches it or not. Can be environmental, let's see that. And the category, I want it to be, let's say, aquatic. And the ecosystem type, here I can choose what I want. I'm going to look for bacteria. And I'm just going to get those studies and see if we find something. Internet gives some problems today. I apologize. You should actually say that there are no results in case there are, it doesn't work. Let me try to reduce that and see if it works better like this. In the meanwhile, I can do a similar search also for, oh, there we go, also for the samples. And here it gives me the same table. It's the same table that I saw before. So the visual is pretty similar to the previous one. Here it tells me basically the different studies that were done in aquatic environment, and for example, it was coral reef and so on, and it was published in PLOS ONE. And I can also find the link if I want to the paper. Oh, this time it works. If I look closer to the study, it gives me lots of more information, where it was published, the NCBI project ID, and the sequencing center, so how it was, where it was done probably, so it was a 454 sequencing. So if we move on, we can look at, if you look at from the main page, we can click on complete projects or incomplete or targeted. If you look at complete, it gives a whole list about what the projects are. And there are different informations about that, and it also gives the NCBI links, like this one. If you look at the bottom left, you can see the highlights and NCBI link. The size of what it was sequenced, the percentage of GSC. Basically, is giving some statistics about the sequencing that was done. Those are all the complete projects. So instead, if I look at the distribution here, project type, sequence status, or phylogenetic, if I click on project type, here it basically shows me how it was distributed between archaea, bacteria, and eukarya. For eukaryotic, actually, it's not showing me anything, so I hope it's not my Java plugin. For archaea, I can see that here is a distribution of how the projects are. So what if it was basically analyzing the genome, if it was analyzing the transcriptome, which is this very small 2%, almost 3% fraction, or if it was already sequenced or uncultured. Most of those are basically analysis of the genome also for bacteria. And here it gives the precise numbers of what they are over the total. Another thing I can do is look at the sequence status. So we know that they contain lots of samples that are complete, some in draft, in progress, and so on. If you look at here, most of them are actually, more than half, at least for bacteria, are in progress. But lots of those for archaeas too, are either in progress or draft. Just a smaller fraction of those are actually complete. And here I'm not sure why I cannot see anything for the eukaryotic. As I said before, a nice feature that I have, it's here, if you see where the mouse is, it's a Google Genome Map, and I can also go on Genome Earth. Here I don't have the plugin, but I just want to show that there is the function too. So at the end, we can also look at the statistics of the data that they have, they compiled. Here you can see that the number of completed projects that are present on the website increased, especially from the early 2000s to 2013. This is actually updated to September 2012, and the paper I've read was from December 2012. Now, the last update that I had was, as you can see here, was actually today. And oh, they just increased the number of genomes. That's actually very interesting because as soon as I started this screencast, it was at 28K, and now it's at 30K. So in the past 10 minutes, they put 2,000 more genomes. That's pretty good. So we can also see the sequencing project by October 11, and the genome projects according to the phylogenetic groups. So bacteria are bacterial, eukarya, and archaea. So we can also look at different other statistics with these pie charts based on the phylogenetic distribution of the bacteria, the findings, and where those were done. For example, Broad Institute, Maryland, or in the rest of the world. And here for specific for archaeal bacteria genomes where they were done around October 2012. So if we go back on the main page here, we can let's look at the phylogenetic page if it opens. Here, based on the phylum, it can give you a distribution of all the phyla, I think are called the plural phylum. And here it says basically how many are those. So if you look at the numbers here, if you count how many we have here, and if you look at the numbers in the paper, we can see that these numbers here are way higher. There are way more phyla in here than there were in the paper. And that's a demonstration of how rapidly the metagenomic field is actually increasing and creating available data for the public. Here we can see basically the distribution of the different phylum, of different phyla, for archaeal, for bacterial phylum, and the eukaryotic phylum. So overall, I think it's a pretty easy to navigate website. It contains lots of useful information if you want then to dig closer into the samples, and is able to give us sequenced genomes from microorganisms that we can use for our own studies. As I said, this is a very useful website. However, I've noticed that there are some redundancies. And if you look here, for example, there is Homo sapiens animal, Homo sapiens down here, and I think we have the same thing. Sorry, we have the same thing also for dog I found, and for cat. So there are some redundancies in the database that probably they didn't check. But this is, for example, is a metadata search and allows you to query, actually to search and see the summary of the data based on those different categories that they use to classify them. So at the end, I hope this helped, and thank you.